Hey guys. Okay, we are going to continue with our discussion of physical evidence. So when we left off, I was talking about how evidence needs to be collected from the crime scene, and hopefully it is evidence that is both tangible and probative. And so not only does that have to be recognized as having evidentiary value, then it has to be collected properly. It has to be packaged properly to reduce contamination and further destruction of the evidence. The chain of custody has to be intact, and then it has to be transported to the lab. So let's pretend that all of that has happened. Okay, you have evidence collected by well-trained, competent, ethical individuals, delivered to the lab where everyone else is also competent, has the proper education and training and ethical. Now, obviously, anytime humans are involved, you can have human error, but let's consider that the system is working how it's supposed to. Okay, so the evidence has been transferred to the lab, and normally it is signed over from law enforcement to a person employed by the crime lab who is called an evidence technician. And then it is brought up to the section of the lab where it is going to be analyzed. And then it is assigned to a forensic scientist in that section, and then they take custody of it. Okay, so that's where we're at. So when forensic scientists examine evidence in the lab, they can examine it in one of two ways. So the first, and this always comes first, okay, is called identification. And so this is simply, what is this item? So for example, if it is a white powder, is it heroin? Is it methamphetamine? Is it cocaine? Okay, just identifying what it is. If the item goes to forensic biology, it could be, what is this red-brown stain? Is it blood? Is there semen identified on sexual assault evidence? If a hair is present, is that hair human or is it from a dog? So identification is really straightforward, and it's just telling you what it is, okay? And some sections, like drug chemistry, all they do is identification, okay? They're just telling you if it is an illegal drug, what it is, and how much is there, how much was recovered, okay? Other sections can take it a step further and do then a comparison analysis, okay? And what this is I basically identifying and then comparing evidence to what we call a known standard, okay? Meaning that a known person or place where this sample was collected from. And then the question that's answered in a comparison test is from where or whom could the evidence sample have originated? Right? So let's say that semen is identified on sexual assault evidence, and then a DNA profile is developed from that semen sample. So now you have a male DNA profile. Then a known DNA standard could be taken from the suspect, and you can compare the two. So you're answering the question, does the DNA from the sexual assault evidence match the DNA profile of the suspect? So, for example, if we talk about the Idaho murders, okay, there was a knife sheath that was recovered from the scene, and they found a male DNA profile on that knife sheath, and then they compared it to the DNA profile of Bryan Kohberger, and it matched. Okay, so that is an example of a comparison test. Okay, another would be, let's take the hair, okay, from the identification test and say that, yep, it's a dog hair. Okay, well now, let's say that you have a victim who was kidnapped, and on the suspect's coat, they find this hair that turns out to be canine. Okay, you could take a known sample of hair from the victim's dog and compare the two. So could the coat, could the dog hair on the suspect's coat, could that have come from the victim's dog? Okay, and that would link potentially the suspect to being in the vicinity of the victim's dog and probably in the victim's place of residence. Okay, so comparison tests take longer. So any section that does comparisons doesn't go through as many cases. Like drug chemistry, like I mentioned, all they do is identification tests. And so a typical drug chemist can do about 150 cases a month. In DNA, which is all comparison, I typically put out about 12 to 15 cases a month. So it's more involved because you have that extra step of doing the actual comparison. Okay, so the big wordy definition of identification tests, if we go back to that, it's basically to take whatever was submitted and then try to determine with as near as absolute scientific certainty what that particular material could be. Okay, so if it's blood, if it's semen, if it is a type of drug, you want to use the most up-to-date, scientifically accepted techniques. You want to make sure that the people doing the analysis have the proper education and training. One thing that people don't think about when someone opens a crime lab is there's usually about a decade worth of work that goes into the science part of opening the crime lab. And we call these validation studies. So basically, if a crime lab is saying, okay, we're going to identify blood, and we can identify blood as human, dog, cat, all the different species, and we can identify it down to this level, then you better have a lot of research that backs that up. You better be trying to take known samples of the different species of blood and then using the instrumentation that you use in the lab to actually identify them at the amounts that you say you can. Okay, so usually these validation studies just fill volumes and volumes and volumes of notebooks, but you have to do this for every single section. Basically, whatever you say you can do in the lab, you have to back it up with research that proves you can do that, okay? And proves that your machines are the most up-to-date and that you're getting reliable results. And that can't be done in a day. That takes years and years and years. Okay? Also, you have to use the most up-to-date equipment and methods. And this goes back to a Supreme Court decision that basically stated this, and it kept, the goal of it was to keep people from being declared as expert witnesses who were just, kind of making up science and, using equipment that they had in their garage and maybe doing this to make money and be hired as an expert and say whatever. So now a Supreme Court decision says that, yep, if you're going to call yourself a forensic science lab, you have to use the most up-to-date equipment that exists, and that has been found to be scientifically valid in a lot of different fields. And the goal is to make sure that, you can't have people who are not scientifically trained testify as expert witnesses. I mean, a lot of the equipment we use in the lab, it's incredibly expensive. It's not something that a typical individual is going to be able to afford and then be able to maintain and keep validated. Okay, so that comes from the Supreme Court, and it's just to make sure that forensic scientists are actually scientists. Another thing that has to happen is you have to have the ability to compare your samples to databases. So the definition of a database is it is basically a place where, whatever, if it's chemistry, fingerprints, or whatever, samples are stored, but those have to be samples from known sources with known properties. So if you're working in the drug chemistry section and you're going to run an evidence sample through and say, this is methamphetamine, well, you better have purchased a known methamphetamine sample, which you can buy from a chemical company, and it's put in a form where you couldn't consume it, okay? But you have to be able to prove that, yep, if this matches methamphetamine in our database, I can confidently say that yes, it actually is methamphetamine. Okay, when we would test for human blood in the serology section, we would use either, blood from our own bodies, which we knew was blood, or we would get samples, say, from the American Red Cross blood bank, because we knew that it's from a source where we can definitely say that, yep, this is blood. Okay, so you have to have validation studies, you have to have the newest equipment, and you have to have databases, and you have to have people who are properly educated and trained. And that's just to do the identification tests, okay, just saying what it is. And you have to use proper controls when conducting testing. And controls are in any type of science, whether it's someone studying a new cancer drug, or even like a new dietary supplement that claims to do certain things, anything that is scientifically regulated. If you're doing proper science, you're using proper controls. Okay, so I'll get back to that in a minute, but I just want to mention these are common types of identification tests. So in the serology section, you may have a shirt come in and it has red-brown stains on it, and maybe all the detective wants to know is it blood? Okay, and if it's blood, is it human blood? That would be an example of an identification test. Okay, is the body fluid on the sexual assault kit, is there semen there? Is there saliva? So just identifying a body fluid and not taking it to DNA, that is an identification test. If you take it to DNA, then it becomes a comparison test. Okay. Is there explosive residue, maybe at the site of a bombing? Okay. At an arson scene, is there any evidence of gasoline being used or some type of accelerant? Is there gunshot residue on a suspect or a victim's hands, which can potentially show if they recently fired a weapon or were very close to someone who did? As I said, drug chemistry, all they do is identification, okay? Just what type of illicit drug is it? In the microscopy trace section, maybe they're going to look at a piece of maybe some plant debris, and they can tell you, yep, this is from a plant, this is the species that it originated from. So they could say, this was, an oak tree. This is a leaf from an oak tree, for example. They're just identifying what it is. They're not comparing it to anything. Okay, so these are common types of identification tests. So I want to go back and just reiterate talking about proper scientific controls, okay? And remember, this is in any type of science. So if you're taking, let's say you're working out at the gym and you want to build more muscle, maybe you take some type of pre-workout and it claims to, yep, this is going to help you build way more muscle. So you want to look into that product and see how it was tested, okay? If the data is just coming from the manufacturer, that's a little suspect because, of course, they want you to buy their product, right? They're going to paint it in the most positive light. So you want to look on the label and see, were there any scientific studies done maybe by a university, okay, or an outside lab that makes it a little more objective. But no matter what the type of science, you should always run proper scientific controls. But I'm going to talk to you about them in terms of forensic testing, okay? So there's 3 different types of controls that we use in forensic testing, and you have to use all 3 of these, okay? And if even one of the 3 is not valid, doesn't work, looks like it's contaminated, then all the results get thrown out. They get called invalid, and you have to repeat the testing again. And that can be scary because a lot of times in forensic samples, you might have just one hair, or a tiny speck of blood. So you worry about running out of sample, okay? So we really work hard in the crime lab to make sure that all of the chemicals we use are up to date and that, yep, they're gonna work, okay? But yeah, you occasionally get into an issue where the controls don't work and you have no choice but to call that test invalid when that happens, okay? So here are the 3 different types of controls that a forensic test has to have. So let's say, I'm going to give you the example of we're working in the biology serology section, and a detective submits a knife and it looks like there is a red-brown substance on the blade of the knife, which the detective, thinks is blood. And he or she just wants to know, is this blood? Okay, so when we run that sample, which would be the evidence sample, there would be 3 controls that would have to go with it. Okay, the first one is called the positive control. So this is something that should give us, if we use all of our chemicals involved in the test, should give us an expected positive result. So the positive control in this case would be a known blood sample, like something we've gotten from the American Red Cross blood bank, or, one of the scientists has contributed blood, but we know 100% this is blood, and here's the source where it was taken, here's the date it was collected. Okay, so that's what a positive control is. If we're testing for blood, we need to use a substance that we know for sure is blood, and it better give us a positive result or something is wrong with the chemicals we're using. Okay, the opposite of that is a negative control. And this is when we're going to use a known substance that it should give us an expected negative result. So in this case, the example I give you, we should use something that is definitely not blood. So whether we use water or we use maybe a saline solution, like something that might come in an IV bag when you're getting rehydrated, but something that we know definitely is not blood. So this had better give us a negative result. Okay, if it comes up positive, then we have a problem. We know there's contamination somewhere. Okay, so that's the positive and the negative. The other thing that we do, and we specifically do this in DNA, is to run a third control, which we call a manipulation blank. And what this has is it just has the test chemicals that we use, and that's it. So if we run that through our tests and we get a positive result, yikes, that shows us that, yep, we have contamination somewhere. Or if we run this in DNA and a DNA profile shows up in there, then we really worry about contamination. Is it mine? Is it the person who's doing the actual test and somehow I contaminated the sample? But this will show us specifically if the chemicals that we're using to test the evidence are contaminated. And like I said, if any of these don't work, you have to call the testing invalid and rerun it. And, you can bet, throw all those chemicals out, make all the chemicals that you use in the testing fresh, and then test them to make sure that they work, and then rerun the evidence sample if that is possible. Okay, so this is what proper scientific controls are. So then we have comparison tests. Okay, so after, let's say the substance has been identified as blood on the knife, well, now we want to know if that blood could have belonged to the victim. Okay, if the victim was deceased, the medical examiner will have taken a known blood standard from that victim, and we can use that for comparison. So the purpose of a comparison test is to determine whether or not the evidence sample and a known sample or known standard could have had a common source. Okay, so could this DNA profile, does it match the suspect? Meaning it could have originated from the suspect. The DNA profile from the blood on the knife, okay, could that have originated from the victim? So those are comparison tests. So when you hear me talking about an evidence sample, that means that it's whatever was recovered from an item of evidence, whether it's from the crime scene, maybe it's taken from someone at autopsy, maybe it's taken from a suspect, it just depends. But it's something that is collected as an item of evidence in a criminal investigation. Okay, then we use the term known standard. So this is when we collect a sample from a known source, and that source could be a person, and it has a chain of custody where we can say it's definitely from this person, and it was collected on this date. Okay, so let's use the Idaho murders again. So they discovered the knife sheath, right? So the knife sheath is the evidence, and that's where they found the male DNA profile. The known standard would be a DNA sample collected from the suspect, Bryan Kohberger. So detectives, would take a swab and rub that on the inside of his cheek, and they can swear that they are collecting it from Bryan Kohberger. So that's what they're going to put on the chain of custody, say DNA sample collected from Bryan Kohberger on this particular date and time at this location. That is a known standard. So a known standard could also be, fibers from the suspect's sweater, and we're going to compare them to fibers that were found on the victim's body during autopsy. So, it's evidence if it's collected during the criminal investigation, and then if there's a suspect or they want to know a potential source, that is the known standard. And then you're simply going to do the same tests and then compare the two and see if they match, okay? Or if the evidence could have originated from the source of the known standard. Okay, so to properly conduct a comparison test, you have to run the exact same techniques on both the evidence and the known samples. And especially in DNA cases, you always process the evidence before the known standards, okay? Because we don't want to have any chance that say for example, a known person's DNA could have made it into the evidence sample, okay? So I would bet that the Idaho Crime Lab, even if Bryan Kohberger were a suspect right away, they still would have processed the knife sheath, gotten that DNA profile, and then put that evidence away, and then process the known standard of Bryan Kohberger, because you don't want any chance that his DNA could have ended up in the evidence sample. Okay, and DNA is so sensitive that, that can happen. So you always make sure you separate the two. And I usually used to separate them, like I would work the evidence 1 week, get it finished, get the DNA profile, and then start on the known standard the following week. So there was no chance whatsoever that I could have contaminated the evidence with the known standard. So that's really important. And then the results are compared to try to exclude the known standard as having contributed to the evidence sample. And I use that wording because, remember, in the American criminal justice system, the defendant is innocent until proven guilty. So what we're trying to do is we're going to do every test that we can to try to exclude that person or that known standard. And then at the end of the day, if it still matches, then it matches. But you do everything possible, every test you can think of, to try to basically prove that person innocent. Okay, so we're going to stop there, okay, because I'm at about 23 minutes, and then we'll pick up talking about probability. Okay, thank you. Bye.